Hello, good afternoon, everybody. Um, I'm not going to go into the too much details, but I'm what like within 15 minutes span of time period. It is very difficult to go through a very big type of documents. So what I'm going to do is just I'm going to summarize the normal routine procedures that we use for the bacterial identification in a routine laboratory that is either the clinical microbiology laboratory or a food microbiology laboratory or industrial or environmental. So these are the some generalized steps that we commonly use to identify the bacteria. So, like especially with the reference to the graduate students or undergraduate students or the medical laboratory experts, whoever is working with the bacteria, they usually go through these all steps, and at various steps, different expertise help us to find out that what type of bacteria are present in our particular specimen. So, like where we start with the bacterial morphology. For example, like me and you, we meet together. The first thing that we recognize is how we look like, what color of my hairs are, or your hairs are, your eyes, what your height is, how much fat you are, or are you thin or slim or so. So same as with the bacteria also. Like when we have to identify the bacteria, the first thing that we look for is their shape, their size, their arrangement, and this is all about the bacterial morphology. These beside these particular shape, size, and arrangement, that sometimes these bacteria are rod in shape, some are round or cocci in shape, some are spiral in shape, some are comma-like in shape. Beside these different shapes, they also, like we say, that they are arranged in different manners. So some are just scattered in the particular field, while some you will see that they are pairs. They are arranged as pairs. Some are present as chains, long or short. Some are present as bunches, just like the grapes. So like these are the things that is helping us, or that provide a guidelines to us, that okay, initially that okay, this is how they look like, so they can be that particular group of bacteria. Now, how you are going to look for these bacterial shapes, their sizes, their arrangements? Definitely you need some tool, because these are the very tiny, very smallest size in organisms that we cannot observe with our naked eyes. So definitely we need some help, and this help is provided by the help of our different microscopes. Now, the very common microscope that we use in our routine microbiology labs is called as compound microscope. What compound microscope is basically? Compound microscopes are the microscopes that are based on two system of lenses. So one system of lenses is close to your eyes, that's why we call it eyepiece, while the other system of lenses is close to the object that we are going to observe. That's why it is also called as objective lens. So now the magnification power of eyepiece and the magnification of the objective lens, when they are multiplied together, they will give you the maximum magnification of a particular microscope. And usually in a general compound microscope that we use in our clinical or environmental or food or other microbiology labs, normally it is up to 2000 to 2500 times magnification that we can usually obtain from this general compound microscope. Source of light in this compound microscope is basically light, or source of magnification. With the help of this, we can also differentiate between the different parts of the bacterial cell just by having the phase contrast. So just controlling the intensity of the light. So this is the second step that we commonly use for the identification. So with the help of the microscope, we look for the shapes, sizes, and arrangements of these bacteria. Now, here comes another issue, that if you see here, these bacteria can appear in different colors. So most common method of dividing these bacteria into two very large groups at the first point is called Graham's reaction or Graham staining method. So in 1845, there was a scientist named Hans Christian Graham. He, on the basis of the chemical composition of the cell walls of the bacteria, divided them into two big groups and give them the names as gram-positive bacteria and the gram-negative bacteria. And this classification of bacteria or division of bacteria into these two wide groups is still in use, and really we are very much obliged to Hans Christian Graham that he helped us to start this basic differentiation from the first point of Graham's reaction. Now, what happened in this Graham's reaction? Basically, the bacteria commonly we come encounter with, they have the variety of different chemicals present in their cell wall, but majority of bacteria, they have either the maximum of the peptidoglycan contents in their cell wall, or they may have the majority of the lipid contents. So those bacteria that have majority of the peptidoglycan content in their cell wall, they are later on called as gram positive, while those that are rich or they have the largest quantity of the lipid components in their cell walls, they will be called as gram negative bacteria. How we are going to reach to this conclusion that who is gram positive, who is gram negative without looking at them just on the basis of Graham's reaction, then we have to look for a little bit more details. So basically what he did, he used four different stains, that is crystal violet, then he also used iodine, then he used decolorizer, and then the safranine. So what he did, he basically on the bacterial film first apply the crystal violet, that gives the whole film violet color. Then he applied iodine, so iodine get mixed with the crystal violet, and after that this mixture was treated with the iodine alcohol that served as a decolorizer. So what happened that when these bacteria that are more enriched in the lipid content, their lipid will dissolve at this stage into the alcohol or the decolorizer. Now what will happen, that those bacteria that are rich in peptidoglycan content, their color will still remain there, while those that are rich in the lipid content, their color will not be there and they will become colorless. So they will have the space now more for the another color or another stain, just like we do the tie and dyeing of the clothes. So when we apply the secondary stain, that is safranine, what will happen is that those bacteria that have become colorless after the action of the decolorizer, they will take up the color of the safranine. As a result, we will have two different color organisms at the end. So one group will have the purple color, while the other group will have the uh, pink or reddish color of safranine. So those bacteria that still retain the color of the crystal violet, it means they have the peptidoglycan that was not dissolved by the decolorizer, and that's the reason they still appears violet. While those bacteria that appears pink or red in color, it shows that they become decolorized at the stage of treatment with the alcohol, and they were rich in lipid content, that's why that lipid get dissolved and they become colorless, and that's how they take up the color of the secondary or counter dye, that is safranine. So at the end of this Graham's reaction, we will have the two broad groups of bacteria, that is gram positive bacteria and gram negative bacteria. All those that appear purple or they are rich in peptidoglycan content, they will be gram positive, and those who are pink or red and they are rich in the lipid content, they will be gram negative. Now after deciding that which one is gram positive, which one is gram negative, this is that first identification step has complete. Now we are going to grow these bacteria. So on the basis of this identification that gram positive or gram negative, and Then on the basis of their further nutritional requirement, we are going to grow them onto different type of bacterial growth media or bacterial culture media. So generally we have different classes or types of these bacterial growth media. We have basic media that provides the basic nutritions for every single type of bacteria. Then we have enhancement media that basically provide the enrichment or enhancement of growth for the bacteria that are pathogenic in nature, while and in the same specimen, if the normal flora of the bacteria of the body or the specimen is present, this normal flora will be inhibited. So basically they will enhance or they will support the growth of the pathogenic bacteria. Then we have enriched media. Enriched media are basically for the growth of the fastidious organism. They have some special or fussy requirements, so we have to add those special nutrients to that particular media so it can help to grow these bacteria into this onto this particular enriched media. Then we have selective media. Selective media, basically the idea is that they make the selection. So only one group of bacteria will grow while the other group of bacteria will not grow. So usually we use it to differentiate between the gram positive and gram negative bacteria. So if for example we are using the media, for example the McConkey's and excuse me, this McConkey's agar is especially designed to support the growth of the gram-negative bacteria. So gram-positive bacteria will not grow on this media. On the other hand, if we use like for example mannitol salt agar, that media is particularly used to grow the gram-positive bacteria, not the gram-negative bacteria. So the selective media will help us to differentiate between that whatever the organism is growing, whether it is gram-positive or gram-negative. Then we have the differential media. So on the basis of this differential media, we can differentiate between different species, different strains, and different groups of bacteria on the basis of utilization of different chemicals that are present in that particular media. Especially we use different type of sugars. So for example, some of the organism that can utilize glucose, some cannot, some can utilize lactose, some cannot, some can utilize xylose, some cannot, some can utilize mannitol, some cannot. So same thing, like we will add these different chemicals or sugars into those media to get the differentiation on the basis of that utilization on that particular media. And finally, after getting this basic idea about the type of organism on the basis of their growth on different type of media, we will go for the antibacterial and antimicrobial susceptibility of those microorganisms on the specific susceptibility media. So normally the susceptibility media that we commonly use includes Muller-Hinton's agar, and we use different methods. For example, we use antibiotic disc diffusion method, that is also called as Kirby and Loss sensitivity method, or we also use the e-test resistive method, especially when we are specific about the minimal inhibitory concentration of the particular antibiotic. So these susceptibility media are also used at the end for the to complete this overall identification process. Now, after the growth on these bacterial culture media, what we do, like we look for that how these bacteria look like. So they are basically grown in groups of the colonies, and the structure of the texture of those colonies is basically is again further helping us in finding or in interpreting or in identification of these different type of bacteria. So these bacterial colonies, for example, if you can see here in this picture, this is a picture of EMB agar, eosine methylene blue agar. On this you can see these spots. These spots, one single spot is a one bacterial colony. So if we will magnify these colonies, we will see that whether they are round, they are small, they are large, they are pinpoint or flat or convex or undulated, or what type of these colonies are. And again, this different texture of the colony help us to find out that what type of organism is growing here. From here, if you see, we also use this special technique of growing these bacteria. This is called four quadrant method, and the purpose of this special way of growing these bacteria is that at the end or in the center of the plate we can have like the particular individual colonies. These individual colonies are further identified on the basis of their texture, their colony characteristics, and also these are picked for the further spot test to check the utilization of different chemicals, production of different chemicals during their growth activities. So most common spot tests that we use are for the production of the catalase enzyme, coagulase enzyme, oxidase enzyme, release enzyme by the different microorganism, because these are the enzymes that make them either pathogenic or non-pathogenic. Beside these spot tests, we also have different other chemical analysis done, and these for like for example we look for the utilization of indotryptophan, we look for the utilization of TSI agar, citrate utilization, ornithine, H2S production, etc. So all these tests are also done by different methods. For example, if you are doing them manually in the lab, we can use API strips, or we can use anterior tubes, or nowadays in big or large scale laboratories we can use different variety of automatic instruments, for example Vitek, and these chemical analysis will help us to find out that which bacteria is able to utilize which type of chemicals, and again it is a great source of information to reach the conclusion. When we say antimicrobial susceptibility testing, Kirby bar and disk diffusion method is one of the common conventional method that we use, in which on the bacterial on the surface of the antibacterial susceptibility media we make a lawn or the consent culture, and on that we set up different antibiotic discs with a specific concentration of antibiotic that is soaked in those or diffused in these discs, and from these discs it is going to diffuse into the surrounding media. And if the organism is not growing in this surrounding media because of the presence of that antibiotic that is coming out of that particular disc, the zone of inhibition will develop, and the diameter of this zone of inhibition will be taken down to note down the specific sensitivity of that particular antibiotic and at that particular concentration against that bacterial culture. uh, Nowadays also, like, we need to know that what is the exact amount of antibiotic that is effective against one type of bacteria culture. For that purpose we need to know the minimal inhibitory concentration of the antibiotics, and that is why we are nowadays like most commonly using this test strip method. So these are basically the nitrocellulose strips. One single strip for one antibiotic has number of like around more than 100 concentrations of same antibiotics. So we can find out that what is the minimum inhibitory concentration of that particular antibiotic against that particular microorganism that we have cultured on that plate. So this is the way we are doing antimicrobial susceptibility testing nowadays, and after finishing that we are going to do the final reporting on the basis of all our results that we observed in our morphology, in our microscopy, via our bacterial growth on different type of media, via different chemical reactions that we observed during our chemical and spot test analysis, and this all will lead us towards finding that okay, which bacteria is growing, and the antimicrobial susceptibility testing will help us to find out that which antibiotic is really effective against that particular type of bacteria that we are going to identify. With this, like, I will try to, in this like I just summarize that what normally we do, and when we need to go into the details of each and every single step of these, there are a lot of things that we can understand, especially with reference to the bacterial growth media, their requirement in different type of culture and growth media, because every single bacteria have variety of other requirements also, their temperature, required temperature, their humidity requirement, their pH requirement. So all their carbon dioxide and oxygen requirements, on the basis of all these minor things, we can easily find out that okay, that what type of bacteria are growing in our specific specimen. Thank you so very much.